recording?
Yes.
Okay. So A and G, amine and guanine, are two ring bases. And this is a purine. T and C have one ring base and are pyrimidine. So, like, A and G can't go in the same DNA strand because that would make 4 rings and it can't fit 4. It only can fit 3. So, A matches with T and C matches with G. So the mRNA has now made a copy of the entire gene, and it's in the nuclear membrane. So it can't get out, but the nuclear membrane has phospholipid heads and bilayers, like so. But they're a lot closer together. So like it's tightly packed, so nothing can get through. But there is doors, like pores that are like doorways for the messenger RNA so that it can make its way out and go to the ribosome, which is where translation happens. In translation, the mRNA codes are translated into amino acids. You have 20 amino acids in your body, 12 you make, 8 you eat. The ribosome, the ribosome looks like this kind of. So this is your ribosome. This is all solid. So your DNA or your mRNA strand comes along, and then it has CZ blah blah blah. And it reads the codons in groups of 3. The start codon is always methionine, which is AUG. So until the ribosome gets to this, so it like cancels that out. Oh look, there's AUG. So it reads this one, and then it reads in groups of 3. Once it starts reading, the transfer RNA, which carries a specific protein, has an anticodon, which is its pair. So these kind of like float around, and each transfer RNA is programmed to only carry a specific amino acid protein. So AUG would be UAC. So this comes down and drops it on here, creates protein. There's a whole bunch of them around, and this does it for the entire gene until it reaches one of the stop codons. The stop codons are UAA, UAG, and UGA. So once it gets to those, the mRNA strand and protein amino acids folds up and is ready for transport. The ribosome then breaks down because it releases the mRNA strand. And the transfer RNA only pick up a specific cell or a specific protein, so you can't it'll drop that one off and then it'll go back and pick up like the same one to drop when next time it shows up on the gene. Pause.
Smile.
Okay, you recording?
Yep.
Okay. Ugh. Okay, so now we're going to move into mutations. There are 2 reasons that DNA can have mutation, even though it's checked like a bazillion times. It can be in the making of the DNA, so it just causes the cell to grow uncontrollably, which can cause cancer, which is very dangerous, or it can just die off and not have any issues. The other reason is environmental issues, like radiation, UV rays, chemicals, that kind of stuff. There are 2 types of mutations. We have point mutation and frame shift mutation. So here we have our DNA strands. So we have right, we're back to DNA, so there's no uracil anymore, we're back to thymine, because in mRNAs there is no thymine. So we have AGC, TTAC. So the pairs TC, TAATG. They won't stop talking.
I'm sorry.
So in point mutation, the DNA is substituted, or like a base is substituted with something else. So like say this is substituted with an A. It creates a completely different amino acid which can completely alter the chain. In frame shift mutation, it's an addition or subtraction of a base. So we could like add a C in here, which would completely screw up how it's read in groups of threes. Or say this was the group of three that was being read. If we got rid of this pair, then now it's going to read in this group, because it's 1, 2, 3. So it screws up how the rest of the line is read. Reading one base wrong will affect how everything else is read, because it's no longer a specific group of three. If the cell issue is in the sex cell and it'll reproduce with every single cell as your cell grows and creates a human. That is everything about protein synthesis, and mutations.
Pause.
Yeah.